Today, Malaysia is the world's largest producer of palm oil, constituting 47% of the world production. Malaysia is also the world's largest exporter, whereby the commodity forms the central backbone of the national economy and making it an important metric to look at when studying the Malaysian economy. But just like any other agricultural crops, oil palm is not spared from the problem of pests. Let's talk about bagworms. Bagworms are the major defoliator pests of oil palm. The three common species of bagworms are Metisa plana, Petroma pendula, and Mahasana corbeti. Defoliation by bagworms adversely affects the productivity of oil palm by reducing both number and size of fruit bunch. Moderate defoliation, 10 to 30% by bagworm, can cause a yield decline of around 30 to 40% over 2 years. The answer is Metisa plana and Petroma pendula. They are one of the most destructive defoliating agents of oil palm in Malaysia. These bagworms are sessile caterpillars that feed on oil palm leaves and use leaf pieces to cover their silken cases. They are actively fed when they are in form of larvae. Typically, the bagworm larvae prefer the upper surface of the leaf for eating and the lower surface for resting and development. So, for this video, let's take a look on what biotechnology can offer and how it was applied in order to control these pests in oil palm plantation. Let's take a look.
A toxin that is fatal to certain herbivorous insects. It makes proteins that are toxic to some insects when eaten, but no others. The proteins are non-toxic to humans because, like all mammals, we cannot activate them. Applications BT was first discovered in 1901 by a Japanese scientist investigating the decline in silkworm moth populations, which he attributed to the root-shaped gram-positive bacterium. In 1911, it was rediscovered by a German scientist, and a solution of crystallized by BT toxins was found to be highly effective against certain crop pests, including the corn borer, corn rootworm, corn earworm, and bollworms. In the United States, the product was first used commercially as an insecticide spray in 1958, and several different strains of the bacteriums are currently used to control for a number of agricultural insect pests and their larvae. In agriculture, BT is a unique bacterium in that it shares a common place with a number of chemical compounds which are used commercially to control insects important to agriculture. The toxins produced by BT has been used as an insecticide spray since the 1920s and is commonly used in organic farming. BT is also the source of the genes used to genetically modify a number of food crops so that they produce the toxin on their own to deter various insect pests. The toxin is lethal to several orders of insects. BT proteins can also be introduced into the crops themselves through genetic engineering. BT crop varieties are engineered to produce a protein toxic to specific insects and are used in areas with high level levels of infestations of the targeted pest.
Act as biological insecticide which react as stomach poison, and the reaction is specific to targeted species. During sporulation, Bacillus thuringiensis produce protein crystal which is dangerous to kill insect. The crystal is only protoxin that if dissolved in the insect stomach, it will transform into shorter polypeptide that is insecticidal. The protein crystals that in nature are insecticidal known as delta endotoxin. Generally, Bacillus thuringiensis crystal in nature are protoxin, but it become toxic because of proteolysis in digestive system of the insect that altered Bt protoxin into shorter polypeptide. Bt protoxin are chemically altered by the alkaline environment inside the gut of the certain insect. The shorter polypeptide are toxic. The active toxin is interact with the epithelium cell in insect midgut. Bt have caused tiny pores form on cell membrane in digestive tract and disrupt the osmotic balancing of the cells. When interrupted, the cell become cluttered and burst. Within minutes of eating Bt, the insect stop feeding. That will occur primarily by starvation in about 2 to 5 days. Number 1. The action of Bacillus thuringiensis is specific to targeted species. Bacillus thuringiensis is a bacterium commonly used as microbial pesticide. Comes in many different strains and subspecies, each of which kills one specific insect or a few closely related insect species. While application of certain chemical insecticide kill the enemies of bagworm more effectively than they did the pest and disturb natural balance, it caused less number of bagworm natural enemies and lead to the outbreak of bagworms. Number 2. Bacillus thuringiensis bioinsecticide is not toxic to human, mammals, and beneficial insects, but chemical insecticide does. Cypermethrin, for example, can cause death to human when exposed orally, while any animal that feeds on the insecticide also danger towards its toxicity. And number 3. Bacillus thuringiensis bioinsecticide is less toxic towards environment compared to chemical insecticide because Bacillus thuringiensis decompose more quickly while chemical insecticide have longer residual activity, thus providing greater persistence control under the field condition.
They use drone to spray Bacillus thuringiensis or BT. Since the bagworms are actively feed at the area of foliage, so the BT was sprayed at the foliage. Every 500 ml of BT solution will be mixed with 9 liters of water. The drone can fly 50 liters of mixed BT, with every 1 liter can cover 1.5 hectares. In one day, they can cover from 10 to 50 hectares. The battery of drone can only stand for 10 minutes every time they were flown. The drone will be monitored and will be fly up to 3 meters from the old palm tree to avoid it from crash. Conclude that generally, both BT bioinsecticides and the chemical insecticide has given promising results in controlling the Metisa plana population in the olive palm plantation. The application of the BT bioinsecticide is as good as the application of the chemical insecticide, even though the BT might take longer times to be effective as it needs to ingest first by the insects. However,
It is stated before that the chemical insecticide such as the cypermethrin has high level toxicity compared with the BT, which is more selective, environmental friendly, and also safer to use. As for any biological product, the BT product must be evaluated first for its viability before it can use in a large scale. As for this case, the BT product is proven to be effective as just like the chemical insecticide cypermethrin. I guess that's all for this video. Thank you for watching and bye!